AI tore through the DNA of Britain city by city and found something that rewrites the map. One city sitting right there in England carries a genetic signature that has almost nothing to do with the British Isles. It traces back somewhere older, somewhere that changes everything about how we think British identity was formed. Today, we break down every result and show you exactly which city and why. Understanding why one British city produced an anomalous result requires understanding what British genetics actually looks like at baseline. Britain is not genetically simple. It never was. The island has been invaded, settled, and resettled by distinct population waves so many times that modern British DNA is essentially a layered archive of every group that ever crossed the English Channel or the North Sea. Each wave left a genetic signature. Each signature persists at different rates, depending on how thoroughly the incoming population replaced or mixed with what was already there. The UCL team used a technique called identity by descent analysis, IBD, layered on top of fine-scale population structure mapping. IBD analysis identifies segments of DNA that are identical between two individuals, not because of random chance, but because they descend from a common ancestor within a calculable number of generations. The longer the shared segment, the more recent the common ancestor. When you run IBD analysis across an entire population, you can identify clusters, groups of people who share recent common ancestry with each other at rates significantly higher than chance. Those clusters correspond to population. The team identified 17 distinct genetic clusters across the 47,000 British samples. Most of those clusters mapped predictably onto geography and history. A cluster centered on the Welsh borders, showing high continuity with ancient Brittonic Celtic population. A cluster across East Anglia with strong Anglo-Saxon signal. A cluster in northeast England and coastal Yorkshire showing elevated Viking Scandinavian ancestry. A cluster in southwest England showing remnants of pre-Roman Atlantic Celtic ancestry. Standard British population genetics. Well documented, expected. Then, the analysis hit one city and produced a cluster that did not fit any of the 17 predicted categories. The researchers initially assumed a data processing error. They reran the samples 3 times using 2 independent analytical pipelines. Same result every time. Before revealing which city produced the anomaly, you need to understand the full genetic history of Britain, because that history is far stranger and more violent than most people know. Layer 1. The Mesolithic hunter-gatherer. The first humans to inhabit Britain after the last ice age arrived approximately 12,000 years ago as glaciers retreated northward. These were Western hunter-gatherers, genetically related to populations across Western Europe, carrying Y chromosome haplogroup I2 at high rates, and physically characterized by dark skin, dark hair, and blue eyes. Cheddarman, the most famous British Mesolithic specimen, had this exact profile. These hunter-gatherers occupied Britain for roughly 4,000 years. Then, they were almost entirely replaced. Layer 2, the early European farmers. Around 6,000 years ago, farmers from Anatolia, modern Turkey, began expanding across Europe. They reached Britain approximately 4,000 BCE, and within a few centuries had replaced over 90% of the Mesolithic hunter-gatherer ancestry on the island. These were the people who built Stonehenge in its earliest phase. They introduced cattle, wheat, and permanent settlements. Their genetic signature, haplogroup G2A dominant in male lineages, shows up in Neolithic British remains at over 60% frequency. Then, they were almost entirely replaced. Layer 3, the steppe migrants. Around 2,500 BCE, people from the Pontic-Caspian steppe, the grasslands north of the Black Sea, began expanding westward across Europe. They carried Y chromosome haplogroup R1b at extremely high rates. They spoke Proto-Indo-European, the ancestral language from which Latin, Greek, Germanic, the Celtic, and Sanskrit all descend. They herded cattle, rode horses, and carried a genetic variant for lactase persistence, the ability to digest milk as adults, that would eventually dominate European population. When they arrived in Britain, they did not integrate with the early European farmers. They replaced them. Within a few centuries of the Beaker culture, arriving in Britain around 2,400 BCE, ancient DNA evidence shows that up to 90% of the male lineages in Britain had been replaced by steppe-derived R1b haplogroups. The people who built Stonehenge were genetically erased by the people who completed it. This is the population that becomes the foundation of later Bronze Age and Iron Age Britain, the ancestors of the Celtic-speaking populations that the Romans would eventually encounter. Layer 4, the Roman. Rome occupied Britain from 43 CE to approximately 410 CE, nearly 400 years of military occupation, trade, and cultural contact. Roman auxiliary troops came from across the empire: North Africa, the Levant, the Balkans, the Rhine frontier. Genetic studies of Romano-British cemetery remains show surprising diversity. Individuals buried in Roman York included people with sub-Saharan African ancestry. Individuals in Roman London showed Middle Eastern and Mediterranean ancestry. But the Roman genetic contribution to modern British populations is difficult to detect. The Roman administration withdrew, the military garrisons left, the infrastructure collapsed, and the next wave arrived. Layer 5. The Anglo-Saxon. Starting around 450 CE, migrants from what is now Denmark, northern Germany, and the Netherlands began settling eastern and southern Britain. These were the Anglo-Saxon, Germanic peoples speaking the language that would eventually become English. The scale of this migration has been debated for decades. Ancient DNA has now settled the argument. Studies of early Anglo-Saxon burial sites in eastern England show that incoming migrants and their direct descendants dominated the genetic profile of those communities at rates of 75 to 90%. In some regions, the Romano-British population was replaced almost as thoroughly as the Neolithic farmers had been replaced by steppe migrants 2,000 years earlier. Modern populations in East Anglia, Kent, and the East Midlands show Anglo-Saxon ancestry at rates of 35 to 40%, higher than anywhere else in Europe outside of Scandinavia and northern Germany. Layer 6, the Viking. Norse and Danish Vikings raided, traded with, and eventually settled parts of Britain from the late 8th century through the 11th century. The Danelaw, a legal zone covering roughly the northeastern third of England, was under Danish political control for most of the 9th and 10th centuries. Modern populations in Yorkshire, Lincolnshire, and the East Riding show Scandinavian Y chromosome ancestry at rates of 25 to 35%, comparable to some modern Scandinavian population. Layer 7, the Norman. William the Conqueror's invasion of 1066 brought French-speaking Norman aristocrats who replaced the English landowning class almost entirely. But the Normans were small in number, perhaps 8,000 to 10,000 individuals, in a population of over 1 million. Their genetic contribution to modern British populations is estimated at 2 to 4%. By 1100 CE, the genetic mosaic of Britain was essentially set. Hunter-gatherer traces in the west, Neolithic farmer remnants in the northwest, dominant steppe-derived R1b ancestry everywhere, Anglo-Saxon signal in the east, Viking signal in the northeast, a whisper of Norman French in the aristocratic lineages. That is what British genetics looks like at baseline. One city in the UCL study did not show that pattern at all. The anomalous cluster centered on Liverpool, not concentrated in one neighborhood, not isolated in one demographic group, spread broadly across the population the UCL team sampled, present at rates that made it impossible to attribute to recent migration patterns from the 20th or 21st century. Now, the cluster's defining genetic signature was Y chromosome haplogroup R1b, the steppe-derived haplogroup that dominates all of Britain. But the specific subclade of R1b was wrong for England. English R1b populations are dominated by the subclade R1b-S21, associated with Anglo-Saxon and broader Germanic population. This is the variant that dominates across lowland England, east of the Welsh borders. Liverpool's R1b signature was dominated by R1b-L21, specifically the subclade M222, at rates of 38 to 42% in male samples. M222 has a nickname in genetic genealogy circles. It is called the Irish modal haplotype. It is the most common Y chromosome variant in Ireland, appearing at 20 to 25% across the Irish male population and reaching frequencies of over 40% in counties like Donegal, Mayo, and Sligo in the Irish Northwest. Some researchers have associated it with the historical Uí Néill dynasty, the medieval Irish kings who claimed descent from the legendary Nile of the Nine Hostages, though that specific attribution remains contested. What is not contested is the haplogroup's geographic distribution. It is Irish. It is found at high rates in Scotland's Western Isles. It appears in Wales. It shows up in the Breton populations of northwestern France. It does not appear at these rates anywhere in England, except Liverpool. The mitochondrial data reinforce the picture. Liverpool female samples showed haplogroup H1 and H3 at elevated rates, haplogroups strongly associated with Atlantic Celtic population, highest in Ireland and Western Scotland, and present in Liverpool's sampled population at rates significantly above the English baseline. In the IBD analysis completed the story, Liverpool samples shared significantly more recent common ancestry within the past 10 to 15 generations with populations in County Mayo, County Cork, and County Galway than with populations in Manchester, 35 miles away. Genetically, significant portions of Liverpool's population were more Irish than English, and the data told the researchers exactly when and why. The story the DNA was telling was not mysterious once the researchers recognized the Irish signature. It Between 1845 and 1852, Ireland experienced one of the most catastrophic demographic events in modern European history, the Great Famine. Triggered by potato blight destroying the primary food source of Ireland's rural poor, killed approximately 1 million people, and drove another 1 million to emigrate within the famine years alone. By 1900, emigration had reduced Ireland's population from over 8 million to under 4.5 million. Liverpool was the primary port of departure for Irish emigrants crossing to Britain and onward to North America. Ships departing from Dublin, Cork, and the Western Irish ports docked first at Liverpool. The crossing took less than a day. The fare was cheaper than any transatlantic ticket, and for hundreds of thousands of famine refugees with no money and no resources, Liverpool was not a transit point. It was the destination. By 1851, the peak of famine-driven emigration, Liverpool's population included over 100,000 Irish-born residents. In a city of roughly 375,000 people, that represented approximately 25% of the entire population born in Ireland. A further significant percentage were the British-born children of Irish immigrants. But here's where the genetics add something that census records cannot. The census tells you how many Irish people arrived. The DNA tells you which Irish people arrived and where in Ireland they came from. The M222 haplogroup frequency data in Liverpool's modern population points disproportionately toward Connacht, the western province of Ireland, encompassing counties Galway, Mayo, Roscommon, Leitrim, and Sligo. This was the region that experienced the most severe famine mortality and the highest emigration rates. Connacht's population fell by over 30% between 1841 and 1851. Some rural parishes lost over half their inhabitants. The people who left were disproportionately young, disproportionately male in the early waves. They arrived in Liverpool with nothing, and many of them never left. They settled in the Dockland parishes of Vauxhall, Scotland Road, and the North End. They married each other. They maintained community networks centered on parish churches that served almost exclusively Irish congregations, and their genetics persisted. The UCL study found that the Irish admixture signal in Liverpool showed IBD segment lengths consistent with a founding event approximately 6 to 8 generations ago, placing the primary genetic founding event between 1845 and 1870. The famine is written in Liverpool's DNA. The obvious question is why? Irish immigration to Liverpool was not unique. Irish communities formed in Manchester, Birmingham, London, and Glasgow during the same period. The Irish diaspora spread across every major British industrial city. But when the UCL team ran the same haplogroup analysis on those cities, the Irish genetic signal, while present, did not dominate at the same rates. London's Irish signal registered at 8 to 12% in the sampled population. Manchester showed 11 to 15%. Birmingham showed 6 to 9%. Liverpool showed 28 to 34%. Why did Liverpool preserve the Irish genetic signature so much more strongly than comparable cities? 3 factors explain it. Factor 1, concentration. Irish settlement in Liverpool was not distributed evenly across the city. It was concentrated in specific parishes in the northern Docklands. Scotland Road, known locally as the Scotty Road, became so densely Irish that it functioned effectively as an Irish Catholic enclave for over a century. The concentration meant that marriage within the community was the norm rather than the exception. When you have a concentrated community with strong social and religious boundaries, genetic mixing with outside populations is significantly reduced. The parish records bear this out. Marriage registers from Liverpool's Irish Catholic parishes between 1860 and 1920 show endogamy rates, marriage within the community, exceeding 75%. 3/4 of marriages in these parishes were between individuals both descended from Irish immigrant families. That level of endogamy over 5 to 6 generations is sufficient to maintain a distinct genetic signature, even within a larger host population. Factor 2, volume. Liverpool received Irish immigrants in absolute numbers that no other British city matched proportionally. At peak famine emigration, Liverpool was processing over 300,000 Irish arrivals per year, transient and permanent combined. The permanent settlers represented a founding population large enough that even significant outmarriage would not dilute their genetic contribution below detectable levels within the time frame the UCL study was examining. The founding population was simply too large to disappear. Factor 3, continuous reinforcement. Unlike some immigrant communities that arrived in a single wave and then closed, Liverpool's Irish community received continuous immigration from Ireland throughout the late 19th and early 20th centuries. Economic migrants continued arriving after the famine years. Each new wave reinforced the existing genetic signature rather than allowing it to dilute through outmarriage. The combination of high initial concentration, strong community endogamy, and continuous population reinforcement created conditions for genetic persistence that simply did not exist at the same intensity in other British cities. The Irish famine migration explains the M222 dominance in Liverpool's modern population. But when the UCL team dug deeper into the ancient DNA comparison, looking not at 6 to 8 generations of separation but at 50 to 100 generations, they found something that complicated the simple narrative of Irish famine migrants arriving in an English city. Liverpool sits in the historic county of Lancashire. Lancashire's pre-Roman and Roman-era population was Celtic, Brittonic Celtic, the language group ancestral to modern Welsh, Cornish, and Breton. The Brigantes, the dominant tribal confederation of northern England, may occupied the region around what is now Liverpool, and left behind a population with a genetic profile significantly different from the Anglo-Saxon-dominated lowlands to the south and east. When the UCL team looked at Lancashire populations outside Liverpool, rural communities in the Ribble Valley and the Fylde Coast, they found that the ancient Celtic Brittonic signal persisted at higher rates than in counties to the south and east. Lancashire was never as thoroughly Anglo-Saxonized as East Anglia or Kent. The Anglo-Saxon expansion was primarily coastal and lowland. The Highland and northwestern zones of England retained larger proportions of their pre-Anglo-Saxon Celtic population. Lancashire sits at the edge of that zone. So, the baseline Lancashire population, even before the Irish arrived, carried more ancient Atlantic Celtic ancestry than the English average. The Irish immigrants who settled Liverpool in the 19th century were themselves carrying Atlantic Celtic ancestry. Irish genetic profiles show very high levels of continuity with Bronze Age Atlantic populations, the same ancient population that inhabited Britain before the Anglo-Saxons arrived. What the famine migration effectively did, viewed through the lens of population genetics, was reinforce a Celtic genetic layer in northwestern England that the Anglo-Saxon expansion had thinned but never eliminated. The Irish migrants were not replacing the local population. In genetic terms, they were amplifying a signal that had been present in the region for over 3,000 years. While the Irish signal dominated the anomalous cluster, the UCL study found a secondary anomaly within Liverpool's genetic profile that the researchers initially treated as noise.